Yep. Hi, thanks everyone for sticking around. I'm Moyang. Today I'm going to talk about part of my PhD project, site-specific ammonium toxicity of Seagrass Zostera muelleri. So first I'm going to give a brief introduction and then talk about the field experiment we've done. So Seagrass Zostera muelleri is the only seagrass species that is native to New Zealand, and eutrophication is believed to be one of the most important reasons behind its loss in New Zealand and of course in other parts of the world as well. And however, the resilience of seagrass to sediment eutrophication is believed to be site-specific, and previous studies show that it has to do with the biomass and density of the seagrass, and also the sediment characteristics of the location. So our question is first, what is the threshold nitrogen level for seagrass declining or seagrass degradation in the system, and for different sediment types? And second, will seagrass and muddy site be more resilient to this kind of stress, and why? And third, how will this influence the benthic macrofaunal community structure? So in order to understand these problems, we conducted a field experiment manipulating 4 levels of nutrient level. We in northern Tauranga Harbour, North Island, New Zealand. So we had 3 intertidal sites, which represent various sediment characteristics. So in each site, we did randomized block design, and we have 5 blocks. In each blocks, we have 4 plots, which were then randomly assigned to control and different nutrient treatments. So this is how the fertilizer was done for each plot, and this is the amount of fertilizer that were used in different nutrient treatments. So fertilizer was first done after the baseline survey, and then was refertilizer again after the 6 months of the experiment. We monitored seagrass percent cover and pore water nutrient concentration every 3 months, and other sediment properties, seagrass properties, and benthic macrofauna community composition every 6 months. So here is the result for baseline survey. I plotted the seagrass and sediment properties using PCO. And if we can see that the data points from site 1 are here, the circles and triangles for data from site 2, and squares data from site 3. And if we ignore the x and y axis of the PCO and just have a look at the vectors of the variables, for example, mud percent, pore water, ammonium, phosphate, nitrate and nitrite concentration all increase in this direction, and we know that site 1 actually was the sandy site and got the least got the least pore water nutrient. However, it had the highest rhizome biomass and shoot abundance and percent cover. On the other hand, for muddier site 2 and site 3, in the sediment, there was more organic matter, chlorophyll A content, and in the pore water, there was more nutrient. Site 3 also had higher above ground biomass. What's interesting is that the ratio of above ground biomass to rhizome biomass was significantly higher at site 2 and site 3. And this difference will remain in the later experiment, and we will talk about that later. So this is what happened to the sediment pore water ammonium concentration. A significant increase was detected in all treatment plots. So compare with the control plots, in terms of the site differences, significantly higher ammonia were detected for site 2 and site 3, those muddier sites, in the 3rd, 10th, and 13th months. And to measure the response of seagrass, we define multivariate index of seagrass morphology, including shoot abundance, rhizome biomass, percent cover, above ground biomass, leaf width, and leaf length. And from this PCO, we can see that the direction of decreasing cover, biomass, and shoot abundance is correlated with the direction of increasing pore water ammonium concentration. Obviously, they were not happy for the ammonium toxicity. And another thing we can notice is that these data points from site 2, these triangles, were more clustered, distributed, whereas these data points from site 1 and actually these from site 3 were more widely distributed. And this, all these were also evidenced by Permanova result. So let's take a closer look at the percent cover result of the property, one of the property indices. So yes, they were all declined in nutrient treatments. However, they displayed different patterns of decline at different sites. So at site 1, the declining was obvious from after the first 3 months. However, for site 2, there was no significant difference between control and low treatments throughout the experiment. At site 3, there was a little bit of temporal difference. For example, we see declining in medium and high after 6 months, and for low after 13 months. So combine this with our sediment pore water ammonium concentration. The threshold at site 1 is actually 10 millimole. For site 2 is just under 50 millimole, and for site 3 is, well, roughly about 100 millimole. So we wonder why this different resilience for seagrass from different sediment types. And we propose several hypotheses to explain this. And the first one has to do with the sediment characteristics. So probably higher initial nutrient in the muddier side might indicate the higher adaptability of seagrass to higher nutrient stress, and this might also have to do with the diffusion rate. So probably because sandy sites had higher diffusion rate, so the seagrass in sandy site might have been suffered from more acute and higher nutrient, but we just don't know because of the time frame of the experiment. Another hypothesis we propose is that this has to do with the above ground and rhizome biomass ratio. So as I mentioned for the baseline survey, this difference between sites was actually remade throughout the experiment. So this might suggest that seagrass from site 2 and site 3, those muddy sites, might have higher carbon and energy potential for them to fix the ammonium in the seagrass cell so that they will be more tougher. On the other hand, higher density and biomass of the seagrass at site 1 failed to alleviate its ammonium symptoms. Therefore, we suggested that in future, in evaluating the toxicity, ammonium toxicity potential, we should also include the above ground to rhizome biomass ratio as part of the discussion. So in the end, I'm going to talk a little bit about benthic macrofauna community. So yes, macrofauna community composition was altered significantly, and this alteration became more profound as experimental went on. However, no interesting result was found in other important macrofauna community indices, important species or functional groups. So they apparently they seem to be very resilient to the treatments. So our to the treatments, and our explanation to that is that so seagrass, living seagrass, is not the obligate food source for the macrofauna, and the second, macrofauna as important ecosystem engineer, they can actually regulate the biogeochemical concentrations of the sediment. So in summary, why higher resilience for seagrasses in muddy sediment? So it probably has to do with the higher initial sediment nutrient concentration in the sediment. It probably has to do with the higher above ground to rhizome biomass ratio of the seagrass. And the message to coastal and estuarine management is that so if you have limited resource, you probably want to set priority so that you can regulate the nitrogen runoff to sandy seagrass meadow first. And so for macrofauna community under nitrogen stress, yes, the composition is altered. However, we did see temporary resilience. So I'm thinking further studies with even longer time frame might be useful for understanding these kind of questions. And thanks.
Thank you very much. Any questions?
Yep.
Just a quick one. I'm not very familiar with this type of experiment, but I've always wondered when I hear about this, how much nitrogen actually is getting into the individual seagrass. It's in that regard, it comes to the point where you've got different grain sizes, and you will get different diffusion and leaching rates. So it's the rate of input may change in the actual diffusion of the fertilizer itself into the sediment, or from the sediment to the grass, based on a different particle size. Does that explain or confound in any way the interpretation?
Well, actually, well, I didn't incorporate this part of my experiment, this part of my data into this presentation, but we actually also analyzed the carbon and nitrogen percent of the percent in the seagrass tissues, including rhizome and leaves. And unfortunately, yes, we did see increasing of the nitrogen percent in seagrass tissues, including leaves and rhizome. However, it doesn't seem to differ a lot between different treatments. So that's for the treatments. And for sites, we did see significantly higher carbon to nitrogen ratio, or carbon to nitrogen ratio in the sites where seagrass were more resilient to the nutrient. However, this is more to do with the nitrogen that they absorbed rather than the amount of carbon they have. So, yeah.
It's a difficult one to entangle sometimes.
Yeah, it is. I guess this is a mutual problem for all ecophysiologists. Yeah.
Thank you very much.
Very nice talk.
Yeah.
We did a study for 2 years, a urea and phosphorus in a factorial experiment, and I wondered, do you have any phosphorus measurements for your sediments or NTP ratios in your
Seagrass plant?
tissues,
Yeah.
particularly lymph tissue, because that can tell you, we had a contrasting sandy and muddy environments.
Mm-hmm.
Doing this factorial experiment, and this was with Australia, so it behaves a bit differently.
Yeah.
Zostera, but really clear results showed up with that, and definitely a very poor response to the added urea in sandy sediments, whereas in muddy sediments they quite liked it.
Yeah. So you mean the reason why I'm seeing this is probably because the imbalance of the environment and also the plant.
Well, it's actually
Yeah.
So
Mm.
will give you the nitrogen toxicity.
Yeah, I mean, this is a very good point because we, unfortunately, we actually didn't monitor the nitrogen to phosphorus ratio in the plant, but we did have the pore water ammonium and the phosphate data there. I'm thinking, do you think the ratio is a more important thing rather than the absolute value for phosphate? Yeah, so I'm thinking, yeah.
Move on, we're over time.
Yeah, oh, sorry about that. Yeah, I'm thinking like later I would learn more about the ratio rather than the absolute value because I didn't see very interesting story in the absolute value. Yeah, thanks.